Hello everyone and welcome to today's webinar. My name is Nathan Nee, Senior Science Editor for The Scientist, and I will be moderating our discussion. Today, our speaker Benoit Derrien will share new strategies using enzymatically synthesized DNA for gene construction, with a focus on how these approaches can help researchers overcome long-standing challenges in the field. Before we begin, I'd like to thank DNA Script for sponsoring today's webinar. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speaker will address these during the Q&A session that follows the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsor has provided us with some helpful resources related to today's webinar, and we have posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you have minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we will send you the link for the on-demand version via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speaker. Benoit Derrien, PhD, is a senior application scientist at DNA Script, where he has spent the past 7 years developing gene assembly solutions in the field of synthetic biology. He specializes in navigating complex DNA constructions using methods like Gibson Assembly and polymerase cycling assembly. Prior to joining DNA Script, he held doctoral positions at ETH Zurich and the CNRS IBMP, and earned his doctorate from the Institute of Physicochemical Biology in Paris. Let's make sure everything's up and running from a technical standpoint. All right, Benoit, please take it away.
Okay, so hello everyone, and thanks for joining today for this webinar on gene assembly using our EDS technology. So it is entitled Unlocking the Unattainable in Gene Construction, and the idea is to show you how our DNA production process can help you assemble difficult sequence that is difficult, that is a problem for some genes. Okay, so before I start, I'd like to tell you a bit about the company and also about DNA synthesis. And I like to start with this sentence that is really highlighting how important writing DNA is in the revolution of synthetic biology. So this is a sentence from an article in the New York Times Magazine, already from November 2021. And it's really showing how nowadays, DNA synthesis and writing DNA is a bottleneck for synthesis biology. So actually, DNA synthesis is a technology that has been developed in the early 80s, and it has been improved over the years. And after 40 years now, we are able to assemble sequences, but not as fast as we are able to read DNA, actually. And there are several reasons for that. I would say there's two categories of reason. The first is the limits in performance that are linked to the chemistry. The coupling yield in particular is such as there is a limit in length. But the fact that it's a chemical process that is using harsh solvent is also a problem because it's damaging DNA. So if you go for too long, then you have to find ways to dampen the problem of errors in the DNA. All those constraints linked to the chemistry is also a problem in terms of the production, because as you can see on the picture on the left, definitely producing DNA is not something simple. You need facilities dedicated to that with very controlled atmosphere and environment. And it's also producing a lot of waste and chemicals. So it's something that needs like proper labs. So the idea behind the development of enzymatic DNA synthesis when DNAscript was created in 2014 was really to find a new way to produce DNA that would be cleaner, simpler, and that would help us pushing the boundaries of DNA synthesis, basically by making longer DNA, more complex sequences, of being able to produce faster and in a way that is easier to handle. So the core of our technology at DNAscript are those three components. So first, a highly engineered TdT polymerase. Second, modified nucleotides that have a 3' OH reversely blocked to control the synthesis, and a solid support that has been developed to be compatible with this aqueous-based synthesis. So how those components are working together to produce DNA, it's simple. We have our resin on which are grafted iDNA, our initiator DNA, on which the synthesis will start. So the first phase is elongation, where we add the TdT and the blocked nucleotides for addition. Then we wash to remove TdT and the leftovers of nucleotides. And we proceed with deprotection. And then once it's deprotected, you can wash again and start a new cycle. And you're repeating that basically as many times as you need to get the oligo of your length, of your desired length. So basically a cycle is in between 10 and 11 minutes, which is pretty fast for DNA synthesis. And at the end of the process, we have what we call the post-processing process, the post-synthesis process, which is mainly desalting, UV quantification, normalization, and it takes about 3 hours on our platform. So basically, if you're printing an 80 nucleotides oligo, it takes 17 hours and up to 72 hours for our longest oligo that is commercially available, 500 nucleotides. So our DNA has been used in, and we've demonstrated actually the quality of our DNA in many different molecular biology applications. And the focus of today will be gene assembly, of course. And I will try to show you how you can use them in those applications. So first, gene assembly. Not gene assembly, but gene synthesis. Why is it so important? Actually, with the development of synthetic biology and the tremendous importance that genetic engineering and protein engineering has taken over the past 20, 30 years, there's a big need in terms of gene synthesis and DNA fragment synthesis. The process itself is actually a mix of different technologies and methods, and the idea is from nucleotides to end up with full gene sequences. And for this, of course, you will need oligonucleotides that used to be synthesized using phosphoramidite and that are still mainly synthesized using phosphoramidite chemistry. But we believe that enzymatic DNA has an important role to play in the future. Those oligonucleotides have to be stitched together into gene fragments, mainly using PCR or ligation-based assembly. And when you need to go longer, you have to stitch those fragments together using different methods that can be Golden Gate, Gibson, and many others. So the first genes synthesis company was created and launched in 2005. And since then, it's fairly easy to order your genes, go online and order your genes. And most of the time, you will get them within 1 to 4 weeks. but this is true when the synthesis is possible. And just to illustrate this fact that not all sequences can be synthesized, I like to show you this table where it's basically a benchmark that we run in the company on our set of 9 genes that we are testing routinely. And as you can see here, also we've submitted this gene set to 4 different suppliers. And as you can see here, some of those genes are often refused by those suppliers. So today, I will discuss actually the use of EDS oligos in different processes meant to assemble genes. So mainly EDS oligos and PCA, long EDS oligos and Gibson Assembly. And in the end, I will try to convince you that those long EDS oligos are perfectly suited to assemble complex and difficult sequences to synthesize. So let's start with the first example, which is the use of our oligos from our syntax platform to assemble a 1.7 kb gene, which is hemagglutinin from influenza A virus. So the aim of this project was to show that it's really easy and quick and fast to use those oligos combined with PCA and Gibson Assembly to produce genes. And also a second objective was to show that our capability to produce long oligos of good quality is important, because basically the performance you get with 120 mers is equivalent to what you can get with shorter oligos like 60 mers. So for this project, there were 4 phases. The first one was the bioinformatic design and oligo printing. The second one was the assembly of the blocks by PCA and applying error correction. Third phase, day 3, was the gene assembly itself by assembling the blocks into a vector using Gibson Assembly. And finally, the sequence verification that can take as much as 3 days. So basically, a full gene assembly process within 3 days, 4 or 5 days if you count the sequence verification. So the first thing you have to do when you start a project like this is to basically to break down your gene sequence into blocks, because if you check out the literature and the methods that are published, you will see that the average size of blocks when you're doing those kind of things is roughly 500 base pair. So that's what we did using AnyBuilder tools from New England Biolabs. And then the second part is to design your oligos to produce those blocks. And for this, you have to break down your blocks into oligonucleotides. And for this, we use DNAworks that we think is working fantastic for this. So once you've done that, basically you just have to print them on our syntax or to order them at our service. And with 60 mers oligo, it's basically 48 oligos that can be printed in 12 hours, so an overnight synthesis. With 120 mers, it's a total of 22 oligos, and they can be printed in 24 hours. So the phase 2 is the PCA and the production of the blocks. And basically PCA is a 3 PCR step if you're using error correction. And the first step is basically a PCR on a pool of oligos that constitute your block. But this step won't generate the full length fragment. To get this, you will have to perform a second PCR, adding the border primers. And with this, you're able to generate the fragment at the right size. But of course, this fragment contains errors, the errors that are the same errors that are in your oligos and that can be added at the level of the amplification, the DNA amplification. So to remove part of those errors, what you can do is perform error correction. And for this, there's many possibilities on the market, but most of them are based on cleavage. And you have to denaturate your fragments and to reanneal. And by doing this, you will create mismatches in the sequence that are basically detected by enzymes that will cleave at the sites of the mismatches. And some exonuclease will show the border of the fragments to remove the nucleotides that are implicated in the errors. And then once you've done that, of course, you have to perform a third PCR that will regenerate the fragment at the right size. And we did that for the first block, the second block, and the third block. So if we look now what it looks like on a gel, you can see that PCR1, as I told you, is not generating the fragment at the right size, but rather some discrete fragments, creating a ladder. At the stage of PCR2, you generated the fragment at the right size. Then with the error correction, you're degrading most of the fragments because they are basically cleaved off each time there is a mismatch. And then with the third PCR, you regenerate your fragments. And as you can see, it's working very nicely with 60 mer, but also with 120 mers. So what's going on when you basically take those fragments and clone them into bacteria? So here are the sequencing results. So on the left here, you have a table, a graph that is showing the total number of clones that we picked and sequenced. And in the bars, you have like the total number of perfect clones that we got out of this when you're using error correction or when you're not using error correction. And as you can see, using error correction, you are improving by fourfold the number of perfect clones. So basically, on the middle here, you have like the number of clones you need to pick to be sure to get like a perfect construct. And this is the average number of error per construct, which is roughly something around 1 in 1,000. So as you can see, on those graphs, using 60 mer or 120 mer EDS oligo is roughly the same in terms of the quality of the sequence you get. So it's a great result because it shows that you can use longer oligo, and by using longer oligo, you can have more flexibility in your design, but you can also push a bit the length of the fragment that you can produce, which is helpful when you want to reach longer genes, for instance, 4 kb or even more than that. So basically, an example of this is basically a gene that we've assembled with just as one block. It's not a big gene, but it's still twice as much as a regular PCA fragment. And here it's just 1.1 kb. So we did 2 designs, 94 nucleotides oligo, 120. And then we did the full process of PCA and then assembly into the vector. And then once we sequenced, that's the type of error rate that we obtain, which is basically as good as what you have when you're doing like with shorter fragments. Okay. So as you can see, it's really easy to produce gene fragments, even genes. However, sometimes, and here is an example with a carotid gene, you may have sequences that are difficult to produce and that are not compatible with the error correction process. An example of this is this very highly repeated region that is a nightmare for priming. So, of course, if you do a careful design, you might want to avoid any oligo priming in this area. But in any case, if you perform an error correction step in the process, you will necessarily have some cleavage in this part. And this is often leading to big deletions in your final construct. So sometimes the solution is to recode, but you are not necessarily able to recode because sometimes you really want the sequence that is the sequence of the gene, and there's no way to change it. So in this case, we think that using high-quality long oligonucleotides can be a solution, and I will try to convince you that that's the case. And for this, I will show you examples of how you can directly use our long oligos, so 200 to 500, into Gibson assembly process to introduce difficult sequences. So first, I will start with a description of the performance you can expect from our long oligos when you are using them directly in a Gibson assembly process. So basically what we did is we took our long oligos, we mixed them together with linearized plasmid and submitted them to a Gibson reaction, picked colonies, transformed into E. coli cells, picked colonies, Sanger sequence, and did the statistics to show you what you can expect from those long oligos. So the first thing is that, so I will briefly explain to you those graphs because you will see a lot of them. So basically, in blue here, on the left, you have like the total number of clone we picked, the total number of perfect clone we get. On the right, it's the statistics. So basically, the percentage of perfect clone, and in green, it corresponds actually to the number of clone you need to pick to have 90% chance to get a perfect clone. So as you can see here, with 200 MERS and 300 MERS, you are below 5 clones to pick to get the perfect construct, which is perfectly workable. And as you can see here, with 400 MERS, the statistics are greatly increased in terms of clones you need to pick. Is it linked to the quality of the oligo? Actually, no, not really, at least not fully. But more to the Gibson process that is relying on 5' exonuclease that are creating the overlaps that are priming together to assemble the fragments. And it's working here, generating the overlaps in this region. But to create the overlaps in this region, you need to have the polymerase to fill this gap to allow the 5' exonuclease to start working here. So the longer the oligo is, the longer, the more difficult it is, and then of course it will impact the efficiency of integration. So to overcome this situation, what we've done is that we've tested the same reaction by, but instead of having the oligo alone, we annealed it to what we call a 5' adapter that is supposed to help to allow the 5' exonuclease to act immediately when you mixed all the components of the Gibson reaction together. So with 400 MER, you see that you're immediately increasing the integration rate, and then you obtain like a statistic that tells you that you have to pick around 4 to 7 clones to get, depending on the length of the adapter, that you have to pick to get the perfect construct. With 500 MERS, you're reaching the limit, and here again, it's also a limit linked both to the quality but also to the process of Gibson assembly. But you still reach something that is close to 20% for this sequence. So now I will show you difficult sequences because, of course, this was for the method. So an example of difficult sequence that we managed to assemble pretty efficiently is this 300 base pair nucleotide oligonucleotide. That is basically almost 80% GC, and it's made of this bad repeat that constitutes almost 26% of the full sequence. And if you look at the statistics, using this with a 60 MER adapter or 120 MER adapter, we've tried the sequence in sense and anti-sense, but you see that it's not making a real difference. But doing this, you reach a very good rate of perfect construct. And basically here, you can expect to get the perfect clone with just 4 colony picked. And I insist on the fact that there's no error correction implicated in this. A second example is this longer oligo, which is actually a gene fragment of 420 base pair. And the difficulty here is not in this part, even though there's stretches of C that are not very nice here, but it's rather in this part that each time we try to assemble this another way, it was most of the errors were in this area because of this tandem repeat that had stretches of 5 to 6 Gs in a row around. And by just printing this oligo and proceeding with the direct assembly using adapters, again 60 or 120, we managed to get something around 20%. So very close to the 1 in 10 clone to pick to get a perfect construct. And to finish with an example of difficult sequence, I'd like to tell you about the poly, which is something that is very difficult to get with phosphorylated synthesis. And it's actually something that we're producing routinely in the lab to assemble constructs that are linked to mRNA production. And so sometimes it's really interesting to have like the poly A tail that is encoded in your construct. And it is something that is difficult to produce normally. So here it's to illustrate this, it's just an example of what we sequenced. It's a 150 nucleotide poly A. So it has been segmented in order to be able to sequence it at least up to 150. Because of course poly A are difficult to synthesize, but they are also difficult to sequence. So here are some examples of the difficult sequence that you can achieve, you can synthesize, and you can assemble with EDS. And to finish, I want to show you another possibility in terms of assembly, and that can be helpful to assemble gene fragment that contains such difficult sequence. And it's also a way to show you that you can reach good quality or good performances with long oligos in between 400 and 500. So basically what we tried here is to use long oligos to create a double-stranded fragment that we can then assemble into a Gibson reaction. So it's like a gapless assembly of the oligo. So basically you take your vector of interest, linearized. You have your pooled oligos that have been annealed together, and you submit it to a Gibson reaction. And then we sequence and we get the results. So basically here we have 3 different designs. One with 250 nucleotides oligo, one with 350 nucleotides oligo. So it's the average size, because of course they are not all exactly this size, and one with 450 oligos. So it's basically 7, 6, and 4 oligos. And when we do the picking and the statistics, you see here that with 200 and 300 base pair nucleotides oligo, you can reach almost 30% perfect clone. So 7 clone to pick to get the perfect construct, and with 450 oligos, and there's actually 4 different oligos in this pool. You have to pick less than 10 clones to be sure to get the perfect sequence. So as you can see here, the sequence I'm talking about is actually the sequence from it's like the single chain variable fragments from the CAR-T I initially showed you in the presentation. So I'm reaching the end of the presentation, so it's time to wrap up and to conclude. And as a take-homes, I'd like to tell you that I hope I convinced you that it's really easy to produce genes and gene fragments using PCA Gibson assembly and our EDS oligos. And it's actually very quick, so 3 days. I hope that you also are confident that using the 120 nucleotides oligo is a plus, and because of course it can allow you to have it's easier to tweak your design to adapt them to your sequence, and also to push the length of the blocks that you can produce using this method. Error correction is definitely something you have to go for if you're into gene assembly, and it's really improving the workflow, as it allows you to pick less clones. And when you are facing very difficult sequence, definitely you can use our long oligos. It's really simple, easy, and quick, and you can expect good performances from those primers. So you can get the long oligos as a service. It's in between 200 and 5. It's up to 500 nucleotides actually, and as you can see, as you've seen in the presentation, you can definitely do longer sequences with this, and difficult sequences with this. And if you just need like the 120 and you want to have it on site in your lab, you can use our syntax platform. So with this, I'm finishing this presentation, and I thank you for your attention, and I'm ready to answer your question for the Q&A session. Thank you.
Thank you very much, Benoit, for that wonderful presentation. So without further ado, let's get to the Q&A session. And the first question I want to ask is, what is the largest gene size that you have been able to assemble using these methods?
So I would say the longest fragment was something around 1.3 in just one reaction, 1.3 kb, sorry. And for the largest gene at the moment, it's around 4 to 5 kb. But definitely, I mean, you can, Gibson being, it's easy to assemble like up to 10 fragments. So I would say that you can imagine that if you need to go longer, you can go longer. It will all depend on the number and the length and the complexity of the sequence.
Thank you very much. During the presentation, you mentioned up to 500 base pairs as kind of a not a cutoff point, but kind of a threshold. Does something happen when you get above 500 that makes things more complicated?
So actually, 500 is the maximum we propose as a service, but definitely the technology is able to go much longer, and I would say that in R&D we are doing like 1,000 base pair, no problem. It's just that the price, of course, is kind of expensive at this size, and of course you when you pay this price, you want to have like at least a certain amount of correct sequence. So for now, it's not yet proposed as a, it's not yet commercial, but it can go longer than 500.
Mm-hmm. Do you have any insights on what causes a company to reject the synthesis of a particular gene sequence?
I have no explanation or like detailed explanation. It's just a matter of fact. If you order genes at or gene fragments at other companies, it is quite often if you're asking for difficult sequence, let's say inverted repeats, GC rich, low complexity like poly A, poly G, poly C. It will be difficult for them to produce those sequences, as it is difficult for us in a way. But we have more success on the synthesis of those fragments. And yeah, there is explanation like the coupling rate, also the fact that the longer you go with chemistry, the more as it's using arched reagent. Of course, the quality of the DNA will be degraded over with the number of cycles you're adding. So yeah, let's say that up to 200 is the limit of the chemistry. So that's one explanation why the length is complex for chemistry. And in terms of the difficulties, it's also like the structure that single-stranded DNA can take and can fold during the synthesis. So there is way to overcome those structures and things like this, like the temperature. But yeah, of course, the more you have, the longer you go, the more difficult it is to get DNA.
Thank you very much. We have quite a few questions on the error correction mechanism. So I was just wondering if you could provide a little bit more detail into how you perform error correction.
Yeah, so for now, the error correction I've used for those projects is mainly what you can find commercially. And in this respect, exclusively it is linked to cleavage of mismatches. So errors are occurring randomly during the synthesis of the oligo or during the PCR process that are needed for the assembly. So if you take one molecule among all the molecules that are produced during the gene assembly process, you will have they will be different from one another because of those because the mutation will be present at different position. So the process of the error correction is just denature this DNA and mixing the strands in between all those different molecules. And most of the molecules will be okay in terms of similar in terms of sequence, but if you mix the errors, you will have some mismatches that will occur when you will reanneal all those molecules. And those mismatches are actually sensed by the enzyme, the correction enzyme. It's a bit like a T71 assay when you're checking the quality of your CRISPR edit, and then those enzymes will cleave at the site of the mismatch. And in the error correction mix, there is in addition some enzymes that are showing the side exonucleases, basically to remove the problematic part, the part more than just the base, but like all the bases around the site of the error. And then when you perform so you stop the reaction, and when you perform the third PCR, all the site and most of the site of the mutation will have been digested, and this is absolutely random. So you're generating all sorts of fragments, and using this third step of PCR, you are reconstituting the full length fragment with less error, of course. And as you could have seen during the presentation, is usually you decrease by fourfold the number of mutations you have in the fragments. There is other way to do error correction, but it's more like let's say it's not commercial yet, but there is a way where you deplete the fragment with error. So the idea is the same. So you create mismatches by denaturing and renaturing your molecules, and then instead of cleaving, you will use proteins that will attach to those mismatches, and you do a pull down of the fragment where you have errors, and normally you reduce the number of those errors in the pool of molecules.
Thank you very much. Next question, could you provide any details on how long oligos would perform when producing sequences containing complex secondary structures, such as UTRs, ITRs, or DNA origami aptamers?
So origami aptamers, I can't really tell. We've never done such molecules. However, for UTRs and ITRs, so we have projects around this, and for this it's a bit more complex. And what we're doing is, because they will structure as a single strand, and integrating them is more difficult. However, this can be done by synthesizing the sense and antisense, creating the gene fragment, and then introducing it into the molecule using Gibson can be a way. And doing this, there's no problem introducing those difficult sequences in the construct. It's of course more difficult to sequence, but with nanopore or other methods, you can do it.
Thank you very much. Next question, what are the advantages of this method versus E-block and G-blocks?
I would say advantages. I would say that the fact that you can address difficult sequences. Ordering G-blocks is okay. It works. But I'm not sure that you can get with very difficult sequence a G block that is satisfying. And I'm not sure that you can, or for instance, the 80% GC sequence that I show in the presentation, if you order it as a G block, and there's high chance that they will tell you that they can't produce the sequence. So I would say that if you are looking for a method to assemble difficult sequence like this, you have higher chance of success with our oligos than G blocks. Same for poly-A, for instance.
Thank you very much. Does the method only work for mammalian DNA, or can it be applied to plants as well?
It can. There's no restriction. I would say DNA is DNA. I mean, the sequence is different, but it's still A, T, C, and G. So.
Thank you very much. We have a question here that asks whether you can explain that for certain sequences, the percentage of perfect clones is smaller when you use longer adapters.
This is a tough one, actually, because I would say that we would need to. I mean, not sure it's exactly how to say. Statistically relevant, because I would conclude this if I would do a lot of time the same. I mean, we are doing this, but not in a way that could strictly rule out that the rate of integration is all the time lower with this longer adapter compared to the shorter adapter. But I would say that there's trends that we saw. It's basically that from 200 to 300, you don't need adapters. We did with or without adapters, and we see no difference, no significant difference. With 400 base pair, nucleotides, you need adapters, because otherwise you won't get the best integration rate. And with 500, it's absolutely mandatory. But I would say that it is absolutely mandatory to get longer adapter. With 400, it works with 30 almost as well as with 120. With 500, you need at least 90 base pair.
Thank you very much. Do you have protocols available for the PCA gene assembly method, including the error correction aspect?
Yes. Yeah, we have an app note available on our website, and we also have a short, a detailed protocol, including error correction.
Great, thank you very much. And I think we just want to wrap up with one final question. And we do have a couple of people asking if you could provide some numbers in terms of how much oligos cost per synthesized base pair, and also how much in terms of quantity do you get from the service?
Okay, so about the price, it starts from 11 cents the base. However, it will increase with there will above 120 should be more, and with the complexity of the sequence, it can be more. In terms of the quantity, the amount of DNA that you get, it will be 50 picomole, something like that, and you get it in 20 to 25 microliters. 50 is the minimum.
Okay.
Guaranteed.
Thank you very much, Benoit, again for that presentation and for a very lively Q&A session. We really appreciate having you here with us today.
Thank you.
In closing, if anyone has any further questions, please consider reaching out to our speaker directly with their contact information shown on the screen. As a reminder, this webinar will be archived on the scientist's website, and you all receive an email notifying you when the on-demand version becomes available for viewing. Please also make sure to follow the scientist on social media for updates on future webinars and other events. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientist, I'd like to extend a final thanks to our speaker, Benoit Derrien, as well as our sponsor, DNA Script. Thank you, everyone, and have a pleasant day.